Hello, am I audible?
Yeah, ma'am, you're audible.
Yeah, okay, we'll start. So once I start the PowerPoint, let me know if you are able to see the slides and then if my voice is still
Yes,
audible.
ma'am. I can see that slide.
Okay, okay, great. Okay, I hope my slides are visible and my voice is audible. If not, at any time during the session, you can let me know, okay, if there is an issue at any time. So welcome to host Pathogen Interactions.
Okay, ma'am. Okay.
Yeah, thank you. Week 6 live session. I'm one of the TAs, Sumati Raviraj from University of Hyderabad, and the course instructor for this is Professor Himanshu Kumar from ICER. So this week, uh, the syllabus contained of pattern recognition receptors, various signaling pathways in it. Just a second. Yeah, so mainly RLR and NLR signaling pathways, and then RLR associated with diseases and therapeutics, NLR and its diseases, and also discovery of DNA sensor and signaling pathway, and diseases. So I hope you would have all watched the videos related to this. So in this session, we will be going through some topics which will help you in solving your assignment questions. Okay. So the first is LGP2 protein, which is a member of RIG1 like receptors or the RLR family, commonly known as. So this includes RIG1 and MDA5 mainly. So your pathways that will be activated downstream are your RIG1 and MDA5 regulated pathways. So while all three share similarities, that is LGP2, RIG1, MDA5, all are belonging to the RLR family. So all of them are structurally similar to each other, but still function wise, each of them have specific distinct functions and also few similar functions. So each protein might have a different unique function, but all of them are involved in the same pathways. So while all three share structural similarities and are involved in RNA sensing. So all of them are involved in RNA virus sensing, but however, LGP2 has a distinct functional role and regulatory roles. So first is it lacks a card domain. So card is caspase activation and recruitment domains. So unlike RIG1 and MDA5, LGP2 lacks card domain. Card domain mediates the downstream signaling via MAVs, that is mitochondrial antiviral signaling proteins. So your LGP2 lacks this card domain, which will downstream activate the MAVs. As a result, LGP2 cannot trigger interferon responses like RIG1 and MDA5 do. So this is one of the major differences. Then it also has dual regulatory role in antiviral signaling. So positive regulation and negative regulation. Second. Yeah. So positive and negative regulation. So in positive, LGP2 enhances the MDA5 activity by facilitating RNA recognition, and it helps to recognize the RNA and helps in activating MDA5 and filament formation during antiviral signaling. Then negative regulation is it inhibits RIG1 by binding to the viral RNA substrates. So this will prevent the RIG1 activation depending on the the substrate and the entry, viral entry and all conditions. Then RNA binding, uh, but no ATP is activated. So LGP2 retains the DXD domain, that is H-box RNA helicase domain, and like similar to RIG1 and MDF5, which allows it to bind to the viral RNA. However, it can bind, but its ATPase activity is either absent or much weaker when compared to these two. So this affects the ability to translocate on the RNA when compared to RIG1 and MDA5. Then context dependent function. So LGP2 acts as a fine tuner of innate immunity, either by enhancing or by suppressing the antiviral signaling, depending on the virus and the immune response at that context. Then it is particularly critical in MDA5 mediated responses to picornavirus and other long dsRNA viruses. So basically, LGB2 is regulatory RLR that modulates RIG1 and MDA5 rather than directly activating the antiviral response. It lacks card domain, RNA binding function, and context dependent regulatory roles. So this certain in these certain areas, it differs from its other family members like RIG1 and MDA5. So the main things are it lacks card domain, then it has dual regulatory role in antiviral signaling, then RNA binding but no ATPase activity, and it is a fine tuner of innate immunity. However, it directly does not activate the antiviral response. Okay, so next is RIG1 and MDF5 detect the viral DNA and how NF kappa B is involved in this pathway. So your RIG1 and MDF5, as we know, it is part of the RLRs. So they detect the viral RNA and they activate the RLR signaling pathway, leading to the induction of NF kappa B. NF kappa B is nuclear factor kappa light chain enhancer of activated B cells. So this is part of your cytokines. So NF kappa B is directly involved in the RLR pathway after activation of RIG1 and MDF5. So how this happens is first activation of NF-kappa B occurs via MAPS, the mitochondrial antiviral signaling proteins. So RIG1 and MDF5 first interact with MAPS on the mitochondrial outer membrane. So you can see here on the this is the mitochondria, this is your cell, your nucleus, your plasma membrane. This is your cytoplasm, cytosol, and this is your mitochondria. So MAPs are present on your outer membrane of the mitochondria. So you have your RIG1 and MDA5 here. So they interact with MAPs. Then MAPs recruit downstream signaling proteins TRAF3, TRAF6, TBK1, that is I kappa K alpha, epsilon, which are the major players in activating the transcription factors. So you have downstream many other proteins that are involved. You have your IRAC, TRAC4, TRAC6, you have your TRAFs, then I kappa K alpha, IKK gamma, IKK beta, P50, P65, everything. Then all these translocate to the nucleus and help in activation of transcription of your interferons, alpha, beta, and also your NF kappa B. So first is activation of NF kappa B via MAPS. Then NF kappa B activation occurs through TRAF6 IKK alpha beta gamma complex as shown here, and then TBK1 IKKE IKK epsilon phosphorylates IRF3 and IRF7 for production of interferons at the end. So next is NF kappa B drives pro-inflammatory cytokine gene expression. So once activated, NF kappa B translocates to the nucleus where it produces the transcription of pro-inflammatory cytokines like TNF alpha, IL-6, IL-1 beta. So this enhances your immune response or activates your immune response. Then it also helps in the transcription of several chemokines like CXCL10, CCL5, etc. So these are chemoattractants which help in recruiting your immune cells. That is, it helps attracting your immune cells to the site of immune response. Then it also uh, activates the transcription of antiviral effectors, that is your interferon stimulating genes called as ISGs, that help in viral clearance. So you have your RIG1, MDF5 in inactive form and active forms. Then they interact with MAVs. Once they interact with MAVs, then again downstream several other protein interactions start. Your IRAC, TRAC, then your TBK, IKK, TRAF, etc. Then finally, all these proteins, the IRF3, IRF7, phosphorylated, and your P60, P65, all these enter and translocate to the nucleus and start the transcription of the required cytokines, chemokines, and antiviral effector proteins. So finally, in the end, we have So finally, in the end, we have production of interferons, alpha, beta, and other pro-inflammatory cytokines like TNF alpha, IL-6, etc. And also expression of ISGs, which are interferon stimulated genes. So this help in clearance of your virus. Then they also have synergistic roles, your IRFs. So synergistic role with IRF3 and IRF7 in type 1 interferon production. So as we saw here with IRF3, IRF7, which is phosphorylated. So via this also type 1 interferons are produced, and NF-kappa B enhances the antiviral response by co-regulating ISGs and amplifying the immune response. So it not only has a synergistic role with IRF3, 7 in transcription of type 1 interferons, but also by co-regulation of ISGs. Then also regulation of the apoptosis in virus infected cells. So first virus clearance occurs. So if the viral infection is overload and the cell has to be now eliminated because now the cell can't survive. NF-kappa B also regulates the cell survival or apoptosis. So helping eliminate virus infected cells if the immune response fails to control the infection. So like I said, first it will try to control the infection by clearing the virus, but in certain cases, if in certain cells, if the virus load is too much, which cannot be cleared, then the cell cannot survive anymore because the entire regulatory system is now hijacked by virus for its own reproduction. So now the entire cell has to be eliminated because the cell is not in a good condition. So first it will try to control the infection. If it fails to do that, then it will trigger the apoptotic pathway. So finally, to summarize this, after activation of RIG1 and MDF5, NF-kappa B functions as key transcription factor, which induces pro-inflammatory cytokine production for recruitment of immune cells, that is also including your chemokines. Then enhances your type 1 interferon response by working with IRF3, IRF7, and also regulates the apoptosis of infected cells. So this makes NF-kappa B a critical bridge between viral sensing by RLRs and activation of broader innate immunity and inflammatory responses. So your RIG1, MDF5 eventually activate your NF-kappa B. So first activation is done by interacting with MAPs. Then you have your later adaptive proteins, your TRAF, TRAF6, TBK1, IKK, then phosphorylation of your TBK1, then we have regulation, co-regulation with IRF3, 7, and finally it results in production of pro-inflammatory cytokines, chemokines, antiviral effectors, your interferons, and also helps in cell survival, that is during the infection or uh, removal of the completely infected diseased cells by apoptosis. Hope that is clear. Now, the next, if you have any doubt in this pathway, you can let me know. I If you want me to repeat it or anything, any doubt, you can just let me know. Okay. So we'll go to the next one. Interferon gamma inducible protein 16, that is IFI 16. This is a protein that helps the body fight infections and regulate the cell cycles. So your interferon gamma 16 is found in many tissues, including blood, bone marrow, and lymph nodes. So its major functions are it senses viral DNA in the cytoplasm and nucleus, and then it activates the inflammasome and produces cytokines that help fight infections. It also triggers production of interferon beta, your type 1 interferons. Then it also is involved in regulating hematopoietic differentiation and cell proliferation because it regulates the cell cycles, and it is expressed in both cytoplasm and nucleus, and it is involved in the response to virus infection and cell cycle arrest. So when there is a virus infection, or if there is a cell cycle arrest in either the proliferating cells or in the hematopoietic cells or during the differentiation, in both these conditions your IFI6 is involved. Then IFI6, sorry, 16. Your IFI16 is also involved in anti-inflammatory activity. So you can see here, these are your unstimulated cells, that is your normal healthy cells, and then you have your virus infected cells, and then you have cells in conditions of autoinflammation or autoimmunity. In all the places you can see there is IFI16 that is involved. It is present generally in the nucleus in your healthy cells, and in case of stimulation then it translocates, and you can see it in both nucleus and outside the nucleus in cytosol. Then it starts interacting with other partners, and it operates via again your sting and your IRF pathways. So how this is unique from other DNA sensors. So your IFI16 is a DNA sensor that it detects the double stranded DNA
Ma'am,
present.
can I get your presentation in my mail, ma'am? Shall I
Yeah,
send my?
it will be posted, right? All the presentations are uploaded in the drive. So after
Oh,
the class is done, you can download it from there.
ma'am, in the past session, I couldn't unable to attend that sessions, ma'am.
Yeah, everything is there. All the presentations are still available. You can access and you can download them.
Thank you so much, ma'am. Thank
No
you.
problem. Yeah.
Ma'am, how long it will take?
So this is a 2 hour class. So I will
Okay.
mostly finish it in probably 1.5 hour.
Okay, ma'am. Okay.
Okay. Yeah. So where were we? Yeah, so it is a DNA sensor. So, but it has certain unique properties when compared to other DNA sensors like AIM2 and C-gas, because it can sense both DNA and RNA. So, your C-gas, your AIM2, your I516, your C-gas, all these are DNA sensors. Your C-gas recognizes double-stranded DNA and activates the sting pathway for interferon production. So you can see here there are double-stranded DNAs that are present in the cytosol in your autoinflammatory or autoimmune condition, and obviously during the virus infection there will be release of your viral DNA or RNA depending on the type of virus. So in both the cases, this kind of floating DNA or RNA in the cytoplasm will be detected by your DNA sensors. So your C-gas recognizes double-stranded DNA and then it activates your sting pathway. Then your AIM2 recognizes your cytoplasmic double-stranded DNA, but mainly forms an inflammasome to activate caspase 1. So every sensor has a pathway through which it can activate your immune response. So one can be via production of interferons, and one can be via formation of inflammasome. This is the circular wheel like thing is your inflammasome. So then your caspases will be activated. So then basically it will try to clear the infection or infected cells. So your inflammasome is activated, and then your caspase 1 is activated, leading to IL-1 beta and IL-18 release. So your IFI 16, however, unlike these two, it can recognize both nuclear and cytoplasmic DNA, but there are also some studies that suggest that it may also detect some certain RNA species, particularly in viral infected conditions. So then inducing TBK1 pathway exclusively. So IFI 16 can activate TBK1 via sting, but C gas also does this. So this property is not unique to IFI 16, acting only through mitochondrial signaling. So IFI 16 primarily signals through sting pathway. So this is located in the ER and not in the mitochondria. Mitochondria we have MABs. So MABs is a part of RLR pathways where your RIG1 and MDA5 comes rather than DNA sensing. Then next was specializing in recognizing viral RNA exclusively. So IFI 16 is mainly a DNA sensor, though it may have some, it has a ability to a certain extent to recognize RNA, but it is not exclusively RNA. It is majorly a DNA sensor only, but it can also recognize RNA. So that is its unique thing. Then, however, RNA sensing is primarily the role of RIG1 MDF5 TLRs and not IFI 16. So for RNA viruses, we have here RIG1 RLRs basically and TLR. So triggering inflammasome formation directly. This was by AIM2, like we saw here. AIM2 recognizes the DNA and it triggers the formation of inflammasome, not IFI 16. So its primary inflammasome forming DNA sensor is AIM2. IFI can contribute to the formation or contribute to the activation of inflammasome, but it does not help in formation of the inflammasomes like AIM2 does. So basically, you have three DNA sensors that was given there, which was AIM2, CCAS, and you have your IFI 16. So out of these, we major things to remember: CCAS recognizes double-stranded DNA. AIM2 also recognizes double-stranded DNA in cytoplasm, but it also helps in formation of inflammasome. And IFI, it recognizes both DNA and RNA in certain contexts. So it is one of its unique characteristics. Okay, so next is NLRs. So NLR are a family of intracellular pattern recognition receptors. So this play essential roles in innate immunity by detecting microbial components and stress signals. So this pattern recognition is particularly for intracellular because your my innate immunity, it helps in detecting the microbial component and other stress signals which your cell or tissue might be exposed to. So loss of function or mutation in such NLRs can lead to many different kind of inflammatory or immune responses. So majorly, when there is a mutation in NLR, we have reduced inflammatory response. So increased susceptibility to infections. So when your inflammatory response is not to the optimum or not very efficient, obviously you will be more susceptible to infections and your body immunity will be dampened. So, loss of function mutation in NOT2, which is in LLR, impair the recognition of bacterial peptidoglycans. So, this is one of the antigens that has to be detected in order to, eliminate the bacteria that has been infected into the body. But this recognition itself gets impaired because of the loss of function mutation. So then this leads to weakened NF-kappa B signaling, which is required for production of several other chemokines and cytokines for activating immune response. So this will be weakened, and also it increases susceptibility to infections like Crohn's disease and Mycobacterium tuberculosis, that is MTB infections. Then it also leads to defect in inflammasome activation. So impaired IL-1 beta and IL-18 production will be there. So loss of function in NLRP3, a key inflammasome sensor, reduces IL-1 beta and IL-18 secretion, weakening the immune defense against pathogens. Then dysregulated immune homeostasis. So regulation of your immune responses will also change. The balance will be altered. So the autoimmune and inflammatory diseases will arise as a result of this imbalance. So while loss of function mutation can reduce inflammation, they can also cause chronic dysregulation of immune response, contributing to diseases like Crohn's disease or Bloss syndrome. So both of them are not mutations. So this Bloss syndrome is a gain of function mutation. So there are both loss of function mutation and gain of function mutation, and each has a particular type of effect on inflammation. So loss of function mutation, basically in NLRs, that is your NOT-like receptors, particularly in NOT2, are strongly associated with chronic inflammation in GI tract, that is your Crohn's disease. So NOT2 detects bacterial peptidoglycans, like we said, and helps regulate the NF-kappa B and autophagy in the intestinal epithelial cells. And it also results in impaired bacterial sensing because it's unable to detect the peptidoglycans, which leads to dysregulated immune response in the gut, leading to chronic inflammation. So, this is a major genetic risk factor for inflammatory bowel disease, that is IBD, and Crohn's is also one of the IBDs. Then enhancing immune responses against viruses. So loss of function mutations in NLRs reduce immune activation rather than enhancing. So basically your immune activation is reduced because there is weakening of NF-kappa B activation, and then there is no production or reduced production of your interleukins. So it will definitely reduce the immune activation, but enhancing of antiviral responses, not a part of this. Then leading to autoimmune diseases like systemic lupus erythematosus. So SLE is linked to overactive interferon signaling. So this involves C-gas sting or TLR pathways and not NLR pathways loss of function. So NLR loss of function mutation will not majorly affect the SLE or will not cause SLE. SLE is mainly due to overactive interferons, which involves your C-gas sting or TLR pathways, then resulting in immune deficiency syndromes like Bar lymphocytic syndrome. So loss of function mutations in NLRs are not linked with this. Immune deficiencies associated with it are usually partial defects and not complete syndrome. So basically it is not a lymphocytic syndrome, then causing hyperactive immune responses leading to sarcoidosis. So sarcoidosis is a gain of function mutation in NOT2 and not a loss of function. So this leads to excessive granuloma formation. So when there is gain of function mutation in NOT2, then the sarcoidosis occurs, which is granuloma formation. So NLR mutations in human diseases. So the nod-like receptors in intracellular pattern recognition will all, like we discussed, they have both gain of function and loss of function mutations. So every particular gain of function mutation in a different NLR or a loss of function mutation in a different NLR leads to specific kind of disorders or specific kind of deficiencies and immune related diseases. So the basic mutations in NLR genes lead to autoimmune disease, autoinflammatory disease, immune deficiency disorders, etc. So whether they result because of a gain of function mutation or a loss of function mutation is then classified. Then gain of function mutation in NLRs. This leads to excessive activation of the inflammatory pathways because obviously they are helping in the pattern recognition and activation, or stimulating the immune response. When there is gain of function, they themselves are activated, or amplified activation is there. So in turn, or the downstream activation will also be amplified. So there is excessive activation of inflammatory pathways. So this often results in autoinflammatory diseases due to uncontrolled inflammasome activation. So they were also involved in the regulation of these signalings once it's activated. So even when the regulation is also disrupted, then this excessive activation cannot be controlled. And then it will eventually lead to excess production of cytokines, your pro-inflammatory cytokines and other cytokines, which lead to autoinflammatory diseases. So NLRP3 mutation, this is part of your inflammasome. So cryoptorin associated periodic syndromes, CAPS. So NLRP3 mutations lead to this, and also diseases like familial cold, autoinflammatory syndrome, Muckle-Wells syndrome, neonatal onset, multisystem inflammatory disease. So many kinds of autoinflammatory diseases because of NLRP3 mutations. This is gain of function mutation. So the basic mechanism is gain of function in NLRP3 leads to constitutive inflammasome activation. So this means it is continuously activated. So the inflammasome should be only activated when there is a need for it, or when the immune response is required to eliminate certain infection, or if the dams or PAMs are encountered, only then the immune response, it has to be triggered, then your inflammasome should be activated. But when there is gain of function, there is continuous activation of inflammasome, whether or not your PAMs and DAMs are recognized, or it is controlled or uncontrolled. Regardless of this, it will be always active. So this leads to excessive production of IL-1 beta, and then it will lead to systemic inflammation. So you will have symptoms like recurrent fevers, rashes, arthritis, neurological syndrome, because your entire autoimmune, so your entire system will have this auto inflammation that is happening due to the continuous or constitutive inflammasome activation. So basically, the end result is excessive production of interleukin. So the treatment involves several interleukin inhibitors, which try to bring down the level of inflammation. Next is NOD2 mutation. So NOD2 is also an NLR. The gain of function, we discussed, it results in Bloor syndrome. So early onset sarcoidosis. So this is also granuloma. So gain of function leads to NOD2 causing spontaneous activation of NF-kappa B. So this is similar to that, in a way that without a proper stimulus or without a proper trigger, due to the gain of function mutation, spontaneously at any time NF kappa B gets activated, and this leads to excessive formation of granuloma. So granulomatous dermatitis is the symptom, and arthritis, uveitis, eye inflammation, etc. are there. And next is NLRC4 mutation. This also results in autoinflammatory syndrome with enterocolitis. So gain of function mutation of NLRC4 causes again excessive production of interleukins, your IL-18 and IL-1 beta. Then this is severe in case of neonatal enterocolitis and systemic inflammation and macrophage activation syndrome. So all this is eventually very harmful to the cells. So neonatal enterocolitis in the newborns. So this will lead to, auto-inflammatory responses being triggered in the body. Now coming to loss of function mutation. So in loss of function mutations, the immune sensing will be suppressed, or the recognition of your PAMs and DAMs will be weakened. So impairment of immune sensing occurs, and this leads to immune deficiency. Here there is excessive immune response, here there is not enough immune response. So this leads to chronic inflammation or dysregulated immune homeostasis. So same NOD2, when it has a loss of function mutation, it leads to Crohn's disease, that is IBD. Uh, and loss of function mutation leads to impairment of bacterial peptidoglycan, like we discussed, and leading to gut microbiota dysregulation and chronic inflammation. So this gut microbiota dysregulation also occurs. So maybe it was not supposed to, you know, it was supposed to recognize your residential bacteria, or it is trying to, recognize and eliminate any peptidoglycan layer. All these kind of important selective recognition gets impaired. So in these cases, it leads to your gut microbiota dysregulation, which is the major cause of IBD, inflammatory bowel disease. Then chronic gastrointestinal inflammation, diarrhea, abdominal pain. So all these are the basic symptoms of IBD. Then NLRP1 mutation. This leads to susceptibility to autoimmune diseases. So diseases like vitiligo, type 1 diabetes, Addison's disease, all these are also related to your NLRP1 mutations. So loss of function mutation may reduce inflammasome activity, affecting the immune tolerance. So as much as recognition of foreign is important, immune tolerance is also important. So because of loss of function mutation, there is immune tolerance also gets affected. Then NLR mutations, and it also has role in cancer. So NOD2 mutations increase susceptibility to colorectal cancer due to chronic gut inflammation. Then NLRP3 mutations, chronic inflammation may contribute to lung and liver cancers. Then gain of function of NLRs cause autoinflammatory diseases like CAPS and BLAW. And loss of function of NLRs lead to immune deficiencies or chronic inflammation like Crohn's. Then it is also having a major role in cancer due to the chronic inflammation. So this is like a gist table. So for NLRP3, gain of function, these are the diseases associated with this, and eventual effect is IL-1 beta is being produced excessively. Then NOD2, loss of function is Crohn's, NOD2, gain of function is BLAW. So in Crohn's, it is IBD, so impaired bacterial sensing, chronic inflammation of the gut, and BLAW, not to gain of function is granuloma. NLRC4 gain of function is autoinflammatory, again excessive production of interleukins. Then NLRP1 loss of function leads to various conditions like vitiligo, type 1 diabetes, Addison's, etc. And this is due to the reduced inflammasome activation. Then loss of function, gain of function of NOD2 and NLRP3. All of these, all loss of function or gain of function will have uh, a role, or will affect, or will increase the risk for cancers like colorectal cancer, lung cancer, liver cancer, etc. This is a result of chronic inflammation due to the loss of function or gain of function, basically altered function of these genes. So that was about NLRs. And now again the RIG1 protein and its downstream signaling. So RIG1 is a retinoic acid inducible gene, that is a full form. It is a cytosolic RNA sensor that detects virus double-stranded RNA, and it belongs to RIG1 receptor RLR family, that is your a part of your antiviral innate immunity. So then it induces your type 1 interferons for your viral clearance, and also your pro-inflammatory cytokines. So there are several steps in the signaling. We'll go step by step. So step 1 is recognition of the viral RNA. So first it has to detect and it has to recognize the invading antigen, that is your viral RNA. So RIG1 is basically in inactive state due to its repressed conformation. So the activation or inactivation is based on the conformational changes. So a repressed conformation leaves it in inactive state at basal conditions, but upon detecting or upon binding to a viral RNA, RIG1 then undergoes a conformational change, which then leads to its activation. It interacts with K63 linked ubiquitin chains to become activated. So you can see your short double-stranded DNA, long double-stranded DNA, phosphorylated single-stranded DNAs all are present. So your rig1, when it's usually in inactive form, inactive conformation, when it binds to this, it undergoes a conformational change, and then it interacts and gets interacts with the ubiquitin chains and gets activated. So this is your ubiquitin chain. Then next is after interaction, it and getting activated, it interacts with MABs. MABs are present on the mitochondrial outer membrane. So MABs is your primary adapter here. Then activated RIG1 interacts with MABs via card domain. So MABs is anchored to mitochondria, peroxisomes, and mitochondria-associated ER membrane. So MABs is not only present in mitochondria; it's also present in peroxisome and MAM. MAM is mitochondrial-associated ER membrane. So all these organelles present in the cell are not isolated from each other. They have interactions with other organelles and they communicate and connect via connections or tethering, it's called. So one such tethering is your mitochondrial associated ER membrane. So this is the portion where your mitochondria and your ER interact. So if you have a mitochondria like this, you have your ER surrounding your nucleus. So the regions of mitochondria near the ER will interact with each other. They will have tetherings with some proteins like IP3R or VDAC, RYR receptors. And then they will have exchange of calcium. So basically, your mitochondria and ER will have calcium exchange. So these channels are calcium channels, and then it also has several other communications and increase or decrease in MAMS. This might also help transferring other signals. So in same way, your ER also will have connections with the plasma membrane. So then it will interact with the stimulus that comes with from the plasma membrane. There will be interacting proteins of plasma membrane which interact with ER. So when there is a stimulus, it signal transduces to ER, and ER either calcium store or anything opens or activated, or ER stress is triggered, uh, unfolded protein stress is triggered. So basically what I'm trying to say is all these organelles are interconnected. So these maps, so several proteins which are present on one organelle, when the organelle is in interaction with other, it might also be present on those. So in the same way, MABs are majorly anchored to mitochondria, but they are also present in peroxisomes and ER mitochondria connections. Those are mitochondria associated ER membranes. So this ensures robust signal amplification. Then MABs is also known as IPS1, Cardiff, and VISA, that is interferon promoter stimulator, card adapter inducing interferon beta, and then virus induced signaling adapter. So first is to recognize viral RNA. Second is to interact with the MABs, that is for your RIG1. Then uh, one second. Yeah, sorry about that. So the third step is activation of downstream signaling pathways. So MABs, so after recognition of the viral RNA, then activation of RIG1 via ubiquitination and then interacting with MABs. Now there will be downstream activation by MABs. So MABs then recruits multiple signaling proteins leading to activation of two major pathways, that is IRF3, IRF7 for type 1 interferon production, and NF-kappa B, that is inflammatory cytokine production. So at the end, the responses are similar for most of the signal activations. So then MABs, your adapter proteins differ. So your MABs then activates TBK1 and IKK epsilon. Then TBK1 phosphorylates in a similar way IRF3, 7, leading to the nuclear translocation and activation of transcription. then your interferons are produced. So your type 1 interferons will now be produced to enhance your antiviral defense. And also NF kappa B in a similar way, MAPS activates IKK alpha, IKK beta, IKK gamma, which then leads to phosphorylation and degradation of IKB. This allows the NF kappa B to translocate to nucleus, similarly like we saw before. So now also the NF kappa B gets translocated, and it also starts promoting the transcription factors for pro-inflammatory cytokines. So this will again eventually produce your pro-inflammatory cytokines TNF alpha, IL-6, IL-1 beta. So basically, RIG1 detects viral RNA, binds and activates to MAPS. MAPS then recruits TBK1 and IKK epsilon, and then this activates IRF37, and then MAPS also simultaneously will activate IKK complex to activate NF kappa B signaling, and eventually they help in the production of type 1 interferons for viral clearance and inflammatory cytokines, pro-inflammatory cytokines, and complete antiviral immune response is now initiated. This is the summary of this. So this is also a gist, your key adapters and regulators of your RIG1 signaling. So your MAFs, which is also called as IPS, CARTF, VISA, etc. So your MAF is your main adapter protein, which is present majorly on mitochondria and also on some other connected organelles like peroxisomes, ER, etc. So this MAVS is your major adapter protein. This transduces your RIG1 signaling. Then we also have TBK1 IKK epsilon. This phosphorylates IRF3 and 7 to induce type 1 interferon expression. And then we have NEMO, that is IKK alpha, IKK beta, IKK gamma. This activates NF kappa B signaling. This leads to production of pro-inflammatory cytokines. Then we also have TRAF3 and TRAF6 that are involved here. TRAF3 facilitates the TBK1 IRF activation for interferon induction. TRAF6 is involved in NF kappa B signaling, but it is not a direct RIG1 adapter. Then LGP2, which is an RLR. So this regulates your RIG1 and MDA5 signaling, like we discussed earlier. So this acts like a fine tuner, right? So it enhances or it inhibits the signaling, and it basically kind of balances or regulates it. So other than this, you also have your OTUD5 and RNF125. So they have roles in activation and degradation of RIG1. So deubiquitination of RIG1 that stabilizes it, and then E3 ligase that promotes RIG1 degradation, negative regulation. So activation and then regulation, both are required. So these are major regulators, the bottom three. The top one are all either activators or adapter proteins which facilitate in the activation of the signaling. Next is activation process of RIG1 and MDA5 in RLR pathway. So this is also similar. So your RIG1, MDA5, like we know, are your cytosolic RNA sensors belonging to the RLR family. They detect viral RNA and activate innate immune responses primarily through induction of type 1 interferons and produce pro-inflammatory cytokines. This is what we know till now. Then activation of it, same. Again, viral RNA recognition. So first is your inactive state. Your RIG1, which is present in inactive state, remains auto-repressed, and intramolecular interactions between your CTD and CART domains occurs. So upon recognition, uh, of a virus, that is it's a viral signature. Upon viral infection, it will recognize a short double-stranded RNA or the 5' triphosphate, that is 5' PPP, that is 3 phosphorylations chain, and single-stranded RNA also. So conformational change and ubiquitination we discussed. that is rig 1 undergoes ATP. So in detail, if you want to know, so rig 1 undergoes ATP hydrolysis. This leads to the conformational change from inactive to active states. Then this exposes the card domain of it. So once this card domain is exposed, now K3 K63 linked polyubiquitination occurs. This polyubiquitination is mediated by TRIM25, and this stabilizes rig1 activation. Then MAVS activation. After this, rig1 interacts with MAVS like we discussed. The card-card interaction, card-domain interaction occurs. Then MAVS forms prion-like aggregates on mitochondria, peroxisomes and MAVS. Then downstream, same IRF3/7 pathway. MAPS recruits your TRAF in turn TBK1, IKK, leading to IRF3/7 phosphorylation. Then it translocates to nucleus, induces interferon alpha beta transcription. Then NF kappa B, MAPS recruits TRAF6, IKK, alpha beta, then leading to NF kappa B activation and production of pro-inflammatory cytokines. So MAD5 is also similar. Uh, this was the RIG one we discussed. So MAD5 is also similar. First is viral recognition. This is basic for all. So MAD5 all MDA5 also is in an inactive state. So MDA remains in cytosolic inactive state in the absence of infection. However, during viral infection, MDA5 also it recognizes double-stranded RNA forming filamentous oligomers along the RNA strand. So it forms a filament along with the RNA that it has recognized. So then ATP dependent filament formation occurs. So unlike RIG1, where ubiquitination helps in its activation, MDA5 does not require ubiquitination. MDA5 filaments act as a scaffold, enhancing the MAPS recruit. So since they form the filamentous oligomers, they then form a scaffold which will help in MAPS recruitment. So this also then binds to MAPS and forms MAPS signalosomes. So these are all like uh, small aggregates that they form, and after that TRAF3, TRAF6 similar activation of IRF3/7 and NF kappa B occurs. So then in the induction of end is again same, induction of interferon, cytokines, etc. So this is a table of like comparing RIG1 and MDA5. So viral recognition, for viral RNA recognition, RIG1 recognize a short double-stranded RNA and 5' triphosphate ssRNA as signatures, and MDA5 recognizes long double-stranded RNAs. Activation mechanism for RIG1 is ATP dependent, that is ATP hydrolyzed conformational change and ubiquitination. However, MDA5 is ATP dependent filament formation and scaffold formation. Then both of them have adapted proteins as MAPs only, same. After this, everything will be similar for both of them. Then you have kinases, then you have transcription factors, then end is cytokine production. So RIG1 recognizes this and MDA recognizes this. Then RIG1 activation is ubiquitination, MDA is filament formation. Other than that, the downstream pathway are similar. So both of them result in the production of type 1 interferons and inflammatory cytokines for antiviral defense. Then next is function of ASC in inflammasome complex. Do you guys have any doubts till now? Okay, if there are no doubts, we'll continue. So function of ASC in inflammasome complex. ASC is apoptosis associated speck-like protein. So this also has the card domain. It is a critical adapter protein in the inflammasome complex. So this wheel-like structure is your inflammasome complex. It plays ASC plays a central role in assembling and activating the inflammasome, leading to caspase 1 activation, IL-1 beta, IL-1 18 maturation, and leading to pyroptosis. Then structural features of ASC. ASC is a small adaptive protein with two key domains, that is your PYD domain, your pyrin domain, and card domain. So your pyrin domain facilitates interaction with upstream inflammasome sensors like your NLRP3 and AIM2, and your card domain helps in interaction between inflammasome sensors and caspase 1. So you have your ASC, you have your NLRP3, you have your procaspase. So interaction between these leads to the formation of NLRP3 inflammasome complex. So here you can see the interacting domains, the PYD domain in blue, the card domain in green, and sorry, card domain in circular blue, PYD in this light blue. Then also you have your LRR and caspase 1, etc. all together interacting, and then oligomers of that proteins will form a inflammasome complex. Then these domains allow ASC to act as a scaffold protein, forming a large oligomeric structure called ASC specs. So the specs to amplify the inflammasome signaling. Role of ASC in inflammasome activation process. First is inflammasome sensor activation. So PRRs, the pattern recognition receptors like your NLRP3, NLRs, AIM2, RLRs also, NLRC4, pyrin, etc. Detect the PAMPs and DAMPs, your pathogen associated and danger associated patterns. And then upon activation, these sensors undergo conformational changes, exposing their pyrin domains. So now, because the pyrin domains are exposed, it will help in ASC interaction between the NLRP3 and AIM2, forming a large oligomeric spec. So this is ASC oligomerization or spec formation. These ASC specs serve as a platform for caspase 1 recruitment and activation. Then your caspase 1 activation occurs. So the card domain of the ASC binds to the card domain of the procaspase 1, the card-card interaction, facilitating the proximity to induce self-cleavage. Then activation of caspase 1 then cleaves pro IL-1 beta, pro IL-1, IL-18. So these are the uh, immature forms or pre-forms. So then after cleavage, they will be activated or mature. So now once they are cleaved and activated, they will be having that biological function. So then inflammatory response and pyroptosis is triggered. Then mature IL-1 beta and IL-1 18, that is your active IL-1 beta and IL-18, are secreted, triggered inflammation, and immune cell recruitment occurs. Then caspase 1 also cleaves gas dermin. This leads in formation of pores in the membrane, which will then cause pyroptosis, that is inflammatory cell death. So, a gist of this is scaffold proteins bridging inflammasome sensors NLRP3 and AIM2 with caspase 1. Spec formation is formation of large oligomers to amplify the inflammasome signaling. Then caspase 1 activation facilitates autoproteolysis of procaspase 1 into active caspase 1. Then cytokine maturation, the pro forms of your interleukins are now cleaved and active forms are formed. Then this eventually leads to promotion of gas dermin decleavage, leading to cell lysis by pyroptosis. So ASC links inflammasome sensors. ASC forms ASC specs, that is your oligomer, and ASC mediated caspase 1 activation triggers IL-1 beta and IL-18 release, leading to pyroptosis, driving inflammation. This is the inflammasome and how ASC functions in the inflammasome complex. Next is role of IRF3 in DNA sensing and type 1 interferon production. So IRF3 and IRF7 are involved generally. So IRF3 is a key transcription factor that plays a role in transcription of type 1 interferons. So this occurs in response to viral and microbial DNA detection. So your STING pathway is involved here, activation of IRF3 in DNA sensing pathways. So DNA sensors in cytoplasm detect the double-stranded DNA from virus, bacteria, or host damage, even self-DNA damage DNA particles are detected. And this triggers IRF3 activation via STING TBK pathway. So we just discussed the STING pathway, TBK pathway. So your virus entry, your viral DNA or RNA or your double-stranded DNA. So your RNA goes to your MABs again via your RLRs. Then your double-stranded RNA again is again you have specific TLRs for that, and then your double-stranded DNA, uh, your CGAS STING pathway is activated. So you have CGAS, CGAM, STING, then eventually again activates TBK1, and this will lead to phosphorylation of IRF3. So your phosphorylated IRF3 will now translocate into the nucleus, and it will activate or promote the transcription of interferon genes. So this is the basic, and the step by step. First is DNA recognition by cytosolic sensors. So you have your C-gas that binds to double-stranded DNA and produces C-GAMP. And C-GAMP activates META or STING. META is another name for STING. It is a stimulator of interferon genes and ER associated protein. Then next is TBK1, then mediates IRF3 phosphorylation. So STING then after activation recruits and activates TBK1. Then TBK1 is a kinase, so it phosphorylates IRF3, leading to its activation. Then next is after activation, after that is phosphorylation, once phosphorylated, IRF3 will dimerize. So dimerization helps it in translocation to nucleus. So phosphorylated IRF3 dimerizes and translocates into the nucleus. It binds to interferon stimulated response elements and promoters of interferon and interferon stimulated genes, and transcription then begins. So finally, IRF3 induces the transcription of interferon beta and then interferon alpha. Then it also interferon beta secretion also triggers the activation of JAK-STAT signaling, which then leads to expression of other antiviral genes. So this is all about the IRF3. So yeah, so we basically discussed mostly about NLRs, RLRs, and IRFs, and how the pathway gets activated, what is DNA sensor, what it detects, which sensor is different from which sensor, then loss of function, gain of function, mutations, related diseases, etc. So other than this, I hope this should be good enough to help you solve the assignment. Other than this, if you guys have any questions, you can ask me, or any doubts. You can also mail me to this email ID if later, after accessing the PPT, if you have doubts any time later during the week also, you can just mail it, either post it in the NPTEL profile, or you can mail directly to me. I will discuss about it in the next session. I think that's it for this session. We can end it. Thank you.